thank you very much, the organizer, for inviting me here. And especially thanks to you to hold on until the very end. Um, yeah, today I'm going to present you the kind of work I'm doing, I've done and recently, and I'm working on at the moment. And from, as you can guess from my title, is quite a different topic from all of you, from what, well, most of you are working on, but I think it will give you a different perspective of, what you can do with venomous animals. And so let me start by telling you a bit about venom research in the last 20 years. As we can see here, it's really increasing, like the number of publications related to animal venoms have been increasing. And I believe that one of the reasons, well, besides a general increase in interest in this kind of research, it might be also due to the fact that in the last 20 years, there's really been an increase in omics technologies. There has been the like sequencing revolution. And in particular, in the last 10 years, as we can see here, there's really been an exponential increase in papers publishing venom gland transcriptome. And also, I think it's probably due to the fact that now we can really access this technology, even working on non-model systems. And so what are the goals generally of venom gland transcriptomic studies? So the one I think is the main one is to combine the transcriptomic data with the proteomics to help with the identification of the toxin in the mass spectra. So it's really function as a database for proteomic studies. And another reason why we look at the venom gland transcriptome is to get an idea of the composition of the venom, especially from those animals which are very tiny and we can't get really enough venom for traditional proteomics analysis. And then, of course, because from the nucleotide sequence, we can translate the amino acids and we can obtain the sequence and look for potential toxin of interest for biotechnology and drugs and so on. But the toxins actually, so as we can see, the toxins are generally the protagonists of venom research. But actually in the venom gland transcriptome, the toxins are not really the majority. So the largest proportion of a venom gland transcriptome is what I like to call the non-venom transcriptome. And as we can see here, these are two old studies on rattlesnake venom gland transcriptome. Actually, the toxin don't even build up half of the whole transcriptome. And I think that's the reason why most of this information has been, let's say, disregarded. All the non-venom transcriptome has been disregarded is because I think the general assumption is that, well, all the other genes is just secretory machinery needed for the secretion of toxins. And I think this assumption is also due to some sort of technical artifacts. Because if you look at the gene expression of a tissue by itself, it's really not much informative. Because if you look at the function, for instance, of the most highly expressed genes, well, you don't know if actually these genes are also highly expressed in all the other body tissues. And then you end up with very general functional categories like these genes is involved in cellular processes or metabolic processes. It's expressed in cells or in organelles. So it really doesn't give us an idea of what's going on. To really get an idea of which genes are important in this tissue, we really need to compare its expression to the other body parts and look at those genes which are highly upregulated and generally the genes which are specific, which are upregulated in the tissue are the ones that are very important for the function of the organ. And so why should we care about the non-venom transcriptome? Well, first of all, it helps us to understand the molecular mechanisms of toxin secretion. It helps us to understand how toxins are expressed and secreted. And it also helps us to understand how venom production evolves. So how these glands evolve is this new function. And then finally, it also helps us to understand the origins of venom secreting organs, which for most animals, we actually still don't know. And so with all the published data available so far, I've decided to use them and try to answer one simple question. Are venom gland transcriptome between species? We actually didn't know. We know that venom is one of the most extreme examples of convergent evolution. It has repeatedly evolved across the animal tree of life. We have convergence in its function, which is the most predominant for predation or defense. We have convergence in the organs, in the structures of the venom apparatus. As we can see here, for example, from snakes to scorpions, the general organization is similar. We have convergence in the protein families that are recruited as toxins, in their pharmacological activities, and in their molecular targets. But what about gene expression? So does convergence in function correspond to convergence in gene expression profiles? So I've decided to use the publicly available data. I've narrowed down to 20 species, representing 8 independent origins of venom. And these were species for which I had both the venom gland transcriptome, hopefully in replicates and not only one sample, and also other body tissues. And my hypothesis was that, well, because the venom glands evolve independently in all these different animals, their global gene expression profile might actually be different. And so what I found was actually quite the opposite. Here is a principal component analysis, which I produced by combining all these tissues from all the gene expression value of all the tissues from all different species together. And as we can see here in red, we have the venom gland from different species, and they cluster together, meaning that they are more similar to each other than to any other tissue. And so in order to compare all these different species together, I had to narrow down the number of genes to a small subset of genes which are shared, which are orthologs across all species. And by doing this, I might actually, it might hinder possible lineage-specific patterns. So to overcome this issue, I've decided, okay, I'm going to analyze each species separately, do differential expression analysis, look which genes are highly expressed in the venom gland of all different animals, and see what's their function is the same across all animals. And so here is the results represented with this semantic similarity plot is called. So each bubble is a cluster of similar functional terms, so Go terms that have a similar function. Their size is related to the number of terms in that cluster, and the color is how many species were represented in that bubble. And so what I found is that first, most of the upregulated genes across different species were similar and were, well, not much surprisingly involved in secretion. It is the secretory glands, so it makes sense. But what was surprising is that I found highly upregulated genes involved in the endoplasmic reticular stress, the untranslated protein response pathways. And this was quite interesting because it really gives us an idea of the extreme secretory capacity of the venom gland. But most importantly, it tells us that during the evolutionary assembly of the venom apparatus, stress response mechanisms have been repeatedly adopted to cope with this mass production of toxins. And actually, very recently, a paper from Todd Kestel's lab came out. They look at the epigenetic profiles of the regions where venom genes are located, and they found several transcription binding sites for proteins, for transcription factors that are indeed involved in these stress response mechanisms. So it's a good, let's say, it supports this result. And the second set of genes that I found highly upregulated in venom glands, to my surprise, were all involved in tissue development, cellular differentiation, and turnover. And this was, as I said, quite surprising, because in a secretory gland, I wouldn't expect to find so many genes involved in these functions, which are upregulated. And so one hypothesis to to explain these results is that, well, the venom gland has to constantly produce this large amount of toxins, and this causes DNA damage to the cells, and also even complete destruction of the venom secretory cells, especially those which have holocrine secretion. So they burst open to release the venom granules. So the other main function of the venom glands, besides secreting, is actually to constantly repair itself, to continue carry out its main function. And then finally, I've also found high upregulation of genes involved in regulation and signaling. And here, the genes involved in this pathway were actually different between lineages, contrary to the genes involved in secretory functions. And so to conclude, yes, I actually, well, contrary to my original hypothesis, I found convergence also in the venom gland transcriptomes. And while the secretory network seems to be conserved among different species, the way in which the cells communicate and are regulated seems to be more lineage specific. And this is possibly because of the different independent origins of venom. And so I've answered this first question by comparing non-homologous venom glands, so glands, organs with different origins. And then the second question that I'm trying to answering now is, how did venom glands evolve their new function? So how the venom glands specialized and got his gene expression profile. And this is more tricky to answer, because to do this, we would need to compare the venom gland to homologous structure, so to other structure which has a similar origin, that they share an ancestor. And as I mentioned at the beginning, in most lineages, actually, we do not know which are the homologous structure, or there are no homologous structure, since venom systems are evolutionary novelties. And so among all the animals, there's actually one group, which is one of the favorite in venom research, the consnails, that actually they possess the group of the neogastropoda, the clade where the consnails are located. There's other beautiful carnivore snails that possess homologous, or we think they are homologous glands that we can use to compare to the venom gland. And so here we have phylogenetic tree of some species of neogastropod and outgroups. So the most, let's say, ancestral or basal homologous glands are called esophageal glands. These are quite simple glands. And then we have the gland of Leblanc in the Moricidae and other groups. And this gland is more developed, and from morphological analysis, it seems to have some sort of function like storage or absorption function. And then finally, we have the conus, the consnels with the highly developed venom glands. So in collaboration with Maria Vittoria Modica and Nico Puilandre, and thanks to the expedition in New Caledonia from the Natural History Museum in Paris, we collected all these species. We took samples from these glands and other body tissues. And the first question I tried to answer is similarly to my other work, is there any similarity in gene expression? And in this case, is the gene expression of the venom gland more similar to that of these of the homologous glands? And to my surprise, I found that actually, as you can see in this gene expression tree, the venom glands do not seem to cluster with the other homologous glands, but they actually form a nice big group with salivary glands. And the Leblanc glands instead and the esophageal glands seem to be more closely related instead to the esophagus. And so again, here I've combined all the data together, and then I thought, okay, I'm going to analyze each species separately. And I look which genes are highly expressed in the venom gland, in the Leblanc gland, and so on. And I've looked at the shared tissue specific genes. What does it mean? I've asked how many genes in the venom gland, so specific to the venom gland, are, for example, tissue specific also in another species. So how many venom gland specific genes are also specific in the Leblanc gland. And what I found, as you can see in this plot, is that many venom gland specific genes are also, or their orthologs are also tissue specific in the salivary glands, whereas in the Leblanc gland share more tissue specific genes with the esophageal gland and with the esophagus. And so this tells us that generally, the highly upregulated genes are the one related to the function of the organ. So it means that the venom gland has a more similar function as the salivary gland. Again, they're both secretory glands, whereas its homologous structures have somehow a different function. And so how did the venom gland somehow deviate from its ancestral state, which seems to be more related to digestion? Could it be because of gene duplication? So could it be that these venom gland specific genes underwent through family expansion? And so to test this, I've compared the size of the venom gland specific gene families in venomous species, so in the consnels, versus the non-venomous species. And what I found is that here is, in the y-axis is the number of genes in the venom gland specific orthogroups, so the gene families. And I found that yes, in the venomous species, the number of genes per gene family was actually higher. So there has been gene duplication. And so to conclude, these are very preliminary results. I'm still working on it, but we can already conclude that the venom gland transcriptome has diverged from its ancestor and converged more toward functionally similar glands. And that gene family expansion might have contributed to the evolution of the function of the venom gland function. And so my take-home message from this presentation is that, first of all, I hope to have you convinced that the non-venom transcriptome does much more than just housekeeping and secreting toxin. We can really gain lots of information of how venom glands evolve, of how do we get this venom. And that's also the importance of comparing venom glands with other body tissues. So if you only have the venom gland transcriptome, that's not very informative. So for who of you who are going to do some transcriptomics and have some maybe leftover budget, please consider also sequencing other body tissues. Also, many toxins are actually expressed also in other organs. So that's also something interesting to look into. And before the acknowledgement, I would like to draw your attention to the survey that we are conducting within the framework of UEVEN, the European Venom Network. So within my working group, we are conducting this survey to get an idea of what are the needs of our venom researcher community, which tools we are currently using, but mostly what we would like to see develop. Where do you think, which tools and web resources you think are lacking at the moment or you would like to see developed? And especially in our network, the clinician, the clinical aspect of venom research and the antivenom is not very well represented. So I would invite you to sign up to join the network and take the survey, which you can find the link. I've retweeted I tweeted the link recently. So if you go on my Twitter account, you will see the tweet at the top, and you can click on the link. It's take 5 minutes. And with this, I would like to thank, first of all, the funding and the University of Lausanne and the Swiss Institute of Bioinformatics, my co-authors and collaborators, the research group who is hosting me, and of course, you for the attention.
We have some time for questions.
Thank you. I particularly liked your data on comparing different tissues, which is why I will ask your question on something completely different. So it's two related questions. Does gene copy number duplication correlate with increased transcript number? And in the venomic studies, does transcript number show any correlation with proteomic protein levels, quantitative? I just wasn't sure that there was very much quantitative proteomics. And in other fields, sometimes they correlate and sometimes they don't. And knowing which category you're in is kind of crucial for obvious reasons.
Yeah, there was an initial idea that gene copy number would correlate with expression, with toxin expression, with number of transcripts of different transcripts, like transcript variants.
Yeah.
I'm not sure, but for sure with expression. Was in particularly some studies on the myotoxis of the Crotalus dorissus, or where they were showing gene copy number and expression to be related. But then actually now that we have genome information from different rattlesnake species, it was shown that actually there are not that many genes related to these myotoxins. And so actually this doesn't seem to be much of a correlation.
Or it may correlate for some families and not others.
Yes, yes.
Yeah, okay. But it does actually kind of color how you choose to interpret your results, because you can't assume that an increased transcript number actually has any change to the overall function level.
No.
And you're not alone. Lots of other fields have the same difficulty. It's just irritating.
Yeah, also related to Mike's question is a field or a topic that is almost absolutely neglected, which is not the evolution of the genes, but the evolution of the regulatory system that produces the genes. So there are very few, but there are studies on the post-transcriptional regulation of a transcriptome, so that you have species that have different venom composition, but have the same transcriptome composition. So there is post-translational regulation. And in this system, at least my group has reported a couple of papers showing that microRNAs could be involved. But we couldn't follow this, and I don't see any other that are following about this. So the same that you say, if you have enough money, look for other tissues. I always say to the transcriptomic people, look also at the microRNA composition of your transcriptomes. And if you cannot analyze this, at least through the through the network, or through other kind of data sharing, put on for other people that can analyze in the context they are interested. So I think this would be very interesting to do.
Also something to
Well, you just need to divide the total RNA into small and bigger, and analyze differently, and then you get the microRNA set.
You said you wanted the question. Is there another question in the room? Oh, there's one here. And then also we'll get to online questions next. So get yourselves ready online.
Hi, Giulia. Thank you so much for speaking. I remember reading the paper on the bioarchive as a preprint and thinking it was absolutely superb, and I've emailed you about it before. But this is just a question to ask when you talk about the gene duplication. Out of interest, is this just sort of a general that there is this increase, or is there any specific mechanisms like alternative splicing that are causing this drive as well in the cone snails, or?
Well, we can't really know, also because these are de novo transcriptome assembly, so we would need the genome, and so far only the conus ventricosus genome is well, there are some others, but they're not as good as so that I can't know.
Yeah, so sorry, I think I got caught up in the excitement. Thank you very much.
Okay, I think there is a question online. Would you like to unmute and ask? Or maybe it's in the chat. Okay.
Hi, Giulia. It's a wonderful talk. And I wonder if you have compared the glands of snakes, like salivary Duvernois gland and venom gland of snakes, if you have data to compare, and what you think about that. Thank you.
Yeah, I've also included a few, well, there was a rectal gland from the prairie rattlesnake. That was the only salivary glands from venomous snakes. And well, if it was, I don't know, maybe going back, it will take some time. But in the plot I show at the beginning, there were also salivary glands and other glands from other animals. Yeah, the orange one, and they somehow kind of cluster near the venom glands. So also there was the recent paper by also Agnieszka Barua, where they looked, well, they compared the salivary glands of mammals and snakes and so on, and they found similar secretory pathways and so on. So there seems to be really like a convergence between the venom glands and salivary glands. And I was surprised to find something similar in the consnels, because we know that the venom gland in consnels derived from the really from the esophagus, is developed during metamorphosis from the esophagus, which is still related somehow to the oral and digestive system, but it's still quite a different origin. But there's not many data on salivary glands of venomous snakes. That's really, nobody really look into that.
Okay, all right, thank you so much. Thank you for an excellent talk and an excellent way to end the meeting. Okay, so that just leaves me with a few housekeeping announcements. The first is just to thank again my co-organizers. You've seen Rita around, and you've seen Sohail, and we had Eddie Rowan, who sadly couldn't be here. So we thank him as well. And this conference will be happening again next year around the end of August. So we would love to see you back again. And also we were discussing taking a group photograph just outside. It's not that many people left, but for those who stayed all the way to the end, it would still be lovely to have a photograph.